begin transmission. Now, I'm sure you are all very anxious to find out the fate of our miserable biochemist, Dr. Cordecieras, and we will get to that eventually, but we need to learn a little bit more about fishes before we boot Dr. Cordecieras onto the island of snakes. If we do. We do. Spoiler alert, we do. That's what's going to happen at the end. But until then, we need to talk about some fishes. So we were exploring the deep ocean, as we tend to do when we're looking for new animals. And in this one section of the coral reef, we saw a really unusual creature that looks like this. Really amazingly camouflaged, and it's got this little tentacle thing that it waves in front of its face to lure in prey. And then it has one of the fastest bites we've ever measured. And even when we slow it down, we can barely see it. Incredible speed. This fish this is the frogfish, and it's highly camouflaged. It's got skin that has all kinds of growths and tubercles all over it to help it blend into the surroundings. And its pectoral fins are actually kind of, they're very sturdy and strong. I can actually use them to kind of waddle along the surface. But it spends most of its time sedentary, waiting for a passerby. Some species lure them in, as you saw this one do. And then in 6 milliseconds, it can open its mouth 12 times its resting state and engulf its prey. One of the fastest motions in the animal kingdom so far that we've measured. 6 milliseconds. It's almost faster than our eyes can really register it. So just another example of an incredible fish deep in the ocean. And now let's talk about some more incredible fishes. Today we'll be talking about class Actinopterygii, the true ray-finned bony fishes, and this is a class of tremendous diversity containing over 95% of all fish diversity. And so, as you might expect, there is a spectacular array of forms, from the attractive, like this beautiful sea dragon. This is the sea dragon's name is Susan, and Susan is a wonderful sea dragon who enjoys being a female sea dragon, in part because it is the male sea dragons who have to bear the young and then eventually give birth, which is a fact that Susan finds quite delightful. So there are some really attractive animals like the sea dragon. There are also some unconventionally attractive animals like the wolf eel, and this is Jared. He's a wolf eel, and he likes puzzles and cheesecake, and he is a really great listener. He's the type of guy that will let you finish a thought entirely before interrupting, and so I think we should all try to be a little bit more like Jared. But regardless, this is just 2 little examples that show the diversity of forms within class Actinopterygii, the ray-finned fish. Last lecture, we talked about infraphylum Cyclostomata with the hagfish and the lampreys, and then we began our discussion on the gnathostomates, which are going to be the jawed animals, and so they're going to include everything from sharks to snakes to birds to mammals, all the jawed vertebrates. And we talked about class Chondrichthyes, the cartilaginous fish. So today we're going to focus on class Actinopterygii, the fish with true bones, and so they have a bony skeleton. And if you remember the cyclostomates, they lack internal ossification. They don't really have a bony, any kind of bony skeleton. And the cartilaginous fish, they don't have a true bony skeleton, but they do have some ossified cartilage that gives them some structural support. True bony fish in class Actinopterygii also have opercula, and that's a familiar word, right? It means lid. And opercula are little protective bony plates that cover the gills, and they can draw water across the gills through negative pressure. And Actinopterygians also have swim bladders, so they regulate buoyancy by adding or removing gas, primarily oxygen, from a swim bladder. Within class Actinopterygii, there are 2 subclasses, Chondrostii, which is a very small group, and then Neopterygii, which is a very, very large group. The vast majority of fish are in subclass Neopterygii within class Actinopterygii. Here we have two very interesting fish. This one right here is the ice fish, and the ice fish can live in tremendous depths in extremely cold temperatures without freezing. So their blood has a protein in it that's an anti-freeze protein. It prevents their blood from developing ice crystals in it, and that would disrupt them and then lead to their death. So the ice blood fish can withstand extreme cold. Pretty spectacular superpower. This other fish here is not does not really have a superpower. It just has a fantastic name, and its name is the sarcastic fringehead. It's a fairly small fish, but its jaws widen out in the males to make this sarcastic fringehead, and they will stake out little territory and burrows and then guard them. And whenever a rival male comes within distance, they will expand their jowls and measure each other up. Usually the one that's smaller will eventually back down because they don't want to fight and risk injury. But if they're relatively the same size, they will fight for the rights to that territory. And it's just a spectacular name, sarcastic fringehead. Let's take a look at some of the diversity here. We have subclass Chondrostii. As I said earlier, this is a small group primarily composed of paddlefish, like you see here, and sturgeons. Paddlefish are kind of called paddlefish because it looks like they got a big old paddle on their nose. This paddle on their nose is primarily for sensing electrical currents of their prey. These guys live in dark, murky water where their eyesight isn't very useful. So it's much better to rely on the senses coming from their really elongated rostrum than to try to see their prey. Here is the great white sturgeon, and white sturgeon are really quite beautiful creatures, and they live in pretty far north, but their eggs are delicious. Their eggs are used for caviar, so they can lay millions and millions of eggs at once. And so unfortunately on Earth these sturgeons were hunted to extinction because we loved eating their eggs. And these are really the primary groups within subclass Chondrostii, the paddlefish and the sturgeons. What is distinctive about them is that they are non-teleost, and I'll tell you what that means when we talk about teleost fish. Just for now, know that they're not teleost fish. Their scales, focus on their scales. So their scales are ganoid scales, and fish scales, if you think about their skin, they're going to have a dermis and an epidermis. The scales are of dermal origin, so they're going to be little bony plates embedded that grow from the dermis. And then you're going to have a layer of epidermis on top that has some keratinized type of tissue on it for protection. So the bony layer in Chondrostii fish is very thick and kind of diamond shaped. It's like this hard, bony, plated armor. And this is called ganoid scales. They don't really overlap. They kind of interlock and form a nice heavy plate of armor. So these are slow-moving fish. They're not very fast because they're slow-moving, and but they're well protected. So ganoid scales are these kind of interlocking, heavy, bony scales. Very different than the placoid scales of Chondrichthyes. Now, the class we were most interested in is subclass Neopterygii, but within subclass Neopterygii, there are some exceptions to the synapomorphy rule that are going to be a little tricky, so just pay attention. So remember, Chondrostii were characterized by their ganoid scales and them being non-teleost. Well, bowfins and gars, these two peculiar Neopterygian fish, are also characterized by being non-teleost and having ganoid scales. So these two fish are in Neopterygii because of some other skeletal features, but they share scale affinities with the Chondrostii fish. So bowfins, like this guy here, which is primarily a sport fish, and then gars, like the alligator gar right there, These are neopterygian fish, but they share many synapomorphies with chondrostean fish. So just keep that in mind. All right, teleost fish. This is where the diversity is. And I just want you to look at the spectacular, gruesome, terrifying diversity found in subclass Neopterygii. Now, a few of these are reconstructions of very rare deep sea creatures. And of the 4 of these very strange looking animals, 1 of them is not real, but 3 of them are. And I'm not going to tell you which one because they're all crazy, terrifying, strange looking animals. 3 of them are actually real that you could find in the deep sea right now. Well, probably you couldn't find it. They're pretty rare, but eventually you could maybe find them. So teleost fish are incredibly diverse, and the ocean is terrifying. That's the big takeaway here. Freshwater can also be terrifying, but for completely different reasons. These are tiny little fish. They don't look too dangerous, right? Well, these fish are called candiru fish, and the candiru is a fish native to the Amazon rainforest, and it has fins that are modified into little spines, and it seeks out the ammonia waste from the gills as fish breathe, and swims upstream, up the ammonia stream into the fish's gills, where it sticks out its fins, latches onto its host, and then drinks some blood. Now that's bad enough, but rumor has it, there's a little urban myth about these little creatures. By the way, these creatures are related to catfish. So these parasitic catfish, the candiru, if a human is in the water, a human also releases ammonia waste, and the rumor is that if candiru is in the water when you're urinating, it can swim up your urine stream into your urethra and then latch out its spiny fins into your flesh and be very, very difficult to extract. There are very few cases of this actually happening. Maybe none. There's some controversy there, but still it's a terrifying enough thought that it makes people watch what they're doing when they're in the Amazon, which you should be doing anyway, I suppose. And I can't help but show you this guy. This is a common species. This is a flounder. We eat flounders quite often. They're not particularly rare, but it's just a very strange animal. So it lives on, it's a benthic creature, and it relies on camouflage to kind of ambush its prey, which is very similar to how a lot of rays and skates live as well. They kind of burrow into the substrate for camouflage. But rays and skates are dorso-ventrally flattened, which makes a whole lot of sense if you go from a free living form to one that's an ambush predator on the bottom of the ocean. It makes sense to just flatten from top to bottom. Flounders just said, no, we're going to flatten laterally. So this is a laterally compressed fish that is laying on its side on the bottom of the ocean. This is obvious from the shape of its mouth, right? The mouth still has kind of this ancestral shape to it, and you can tell if you cover up one of the eyes with your hands, it looks just kind of like a normal fish on its side. And either eye you cover up, it works. But when you look at both eyes, this becomes a very, very strange creature. So over time, one of the eyes migrated to the other side of its body, so that they're both on the right side of his body. I guess that would be the right side. Yeah, the right side of his body. Very strange creatures for sure, and it shows you that evolution doesn't always follow the expected path or the most parsimonious path at all. It just wants something that's functional. All right, so let's talk about teleost fish, because all of the rest of the animals and all the rest of the fish are teleost fish. So we have some exceptions in Neopterygii that are non-teleost, but the vast majority of Neopterygian fish are called teleost fish. And teleost fish have 4, maybe 5, I forget, 4 or 5 distinct character traits, and we'll go through them one at a time. So first of all, rather than having placoid scales like the chondrichthyes or ganoid scales like the chondrosteans, these Neopterygian teleost fish have cycloid, ctenoid scales or no scales at all. And the cycloid and ctenoid scales are probably what you think of when you think of fish scales. These are overlapping scales, and they don't have a strong bony core to them. They're lighter, more flexible, primarily kind of keratin, and so they're much lighter, which makes the fish swim much more quickly, and then diversify into all these pretty amazing shapes. So you lose some protection, but you gain speed, which is also kind of a type of protection. So it's costs and benefits there. Some creatures like catfish and eels don't have scales at all, and rather their epidermal layer secretes a kind of mucus that helps protect them, helps them glide through the water. Eels are some of the creepiest creatures in the ocean, right? Look at all these teeth on this tiger eel. And then look at, oh, here's an example of cycloid scales in real life. But going back to the creepy eels, look at this guy here. This is a moray eel. And, you know, I know it's just taken at just the right angle to be creepy, but still, that's not something you would like to see in the dark ocean when you're just out scuba diving or swimming by yourself. And did you know that eels have another eel inside of them? So they have two pairs of jaws, which is pretty spectacular. So if you look at an X-ray of a eel eating this little crab, you see its normal set of jaws, but then inside its throat, it has a second, smaller set of jaws that while the bigger jaws are grabbing the prey, the smaller jaws are coming in and chomping it away. This is remarkable. We don't know precisely where the second pair of jaws comes from. It may be modified gill arches. Regardless, eels are some creepy, creepy creatures. So we have cycloid, ctenoid, or absent scales. Secondly, we have homocircle tails rather than the heterocircle tails that we saw on the chondrichthyes. And this one's pretty easy. Homo means the same, so both the dorsal and ventral lobe of the caudal fin is the same. So heterocircle, usually the dorsal lobe is larger than the ventral lobe. Homocircle is the same. We can see this here on this elaborately decorated pufferfish. We can also see this here on this slow-moving gigantic moonfish. And these are really remarkable creatures. And in all the different teleost fish, it's going to be important for you to be able to see the different fins for what they are. So we have here the dorsal fin. Here would be the pelvic fin. What are these little guys here? These little stubby things here? Those are going to be the pectoral fins. And then this is the homocircle tail. See the lobes are the same on top and bottom, dorsal and ventral side. So homocircle tail. Secondly, or thirdly, they have diverse fin morphology. We kind of were seeing this earlier with the seahorse and the eels, and really all the fish I've shown you have incredibly different functions and therefore forms of their fins. Earlier in the semester, we talked about the cave angelfish. These guys use their fins to walk up vertical waterfalls to lay eggs in little cave pools. And so their fins have little suction cups on them. And the pectoral and pelvic fins are roughly the same size and shape. If you contrast that with the stonefish on the right, the stonefish have really unique dorsal fins that have spines that connect to venom glands. So if two things. They're highly camouflaged, so their fin morphology blends itself well into the coral reefs where the stonefish lives. But then it has venom-tipped fins. Some of the most potent venom out there, actually. And so this is a very dangerous thing to step on if you're in the ocean. This one's one of my favorite, the lipstick fish. And its fins are modified into these little walking stalks that it uses to scuttle along the ocean floor. And why are its lips so red? Well, why do you wear lipsticks? Lipsticks. Do you wear lipsticks? Why do you wear lipstick? To attract mates, of course, right? So that in the lipstick fish species, though, it's the males who have bright red lips, and this is kind of a territorial display. and a way for the females to recognize the males. But same kind of function. Very diverse fin morphology, though. Fourthly, and potentially lastly, we'll have to see what's on the next slide to be sure. But fourthly, teleost fish all eat by suction feeding. So they have jaws, but they don't use their jaws to really bite and grasp their prey. Rather, they eat their prey by opening their jaws really, really widely. This is like what we saw with the frogfish earlier. An incredible negative vacuum pressure is created, and food is just slurped into their mouth that way. So suction feeding is a very unique way of eating, different than a shark actively biting and swallowing. This is just opening quickly, negative vacuum pressure, food goes inside. They can do this because they have pretty spectacular upper jaw bones that can extend out, and this is pretty remarkable, right? We don't really consider fish being able to do this, but this allows them to kind of sneak up on their prey, and before their prey really knows they're in range, they are, and then they're dead. Pretty cool. And that's really all we need to know about the neopterygians for now, as far as synapomorphies. So I guess there were just four. So scales, fins, suction feeding, and the fourth one. So we'll move on to class Sarcopterygii. These are the lobe-finned fishes, and they're going to include coelacanths and lungfish primarily. This is another small group, but it is from Sarcopterygii that supposedly tetrapods evolved, or similar ancestors to these. So these fish have kind of fleshy lobe-like fins, and it is from these that, according to evolutionary theory, eventually legs and feet and hands eventually evolved. So Sarcopterygians are kind of the ancestral form of all tetrapods, the lobe-finned fish. And another reason why they're considered ancestral is that rather than having a swim bladder for buoyancy, they often, in coelacanths, I think their swim bladder is kind of filled with fat to provide buoyancy. But in lungfish, as you might have guessed by the name, they have lungs, so they can breathe air pretty remarkably. And some of the African species can survive out of the water for long periods of time and have been seen kind of ambling across from dried up lake to find a fresh piece of water. So lungfish and coelacanths are Sarcopterygians, have lobe fins and fleshy lobe-like fins, and can breathe air. Coelacanths also have diphthcircal tails, which they have an extra little lobe here, and it's kind of triangular at the end rather than flat with the fin coming off the end. So lungfish also have kind of a triangular shaped tail at the end, so that's a unique type of tail called diphthcircal. Here's one of those fascinating lungfish I was talking to you about like 30 seconds ago. All right, so now that we know the classification, we can talk a little bit about physiology. First of all, fish swim. I know that is a shock to you, but fish are well-known swimmers, very, very good at it. And they swim with some special W-shaped muscles called myomeres. And the way they're arranged makes it so that each muscle is attached to several vertebrae at once. And so you have a very fine, flexible control of swimming. So this can lead to some pretty powerful swimmers. The sailfish up here that I've shown you here, they can swim about 65 miles an hour, which in the water is just phenomenal. That's as fast as a cheetah on land. So these are really remarkable animals. Most fish, of course, swim a lot slower than that, but some of the large swordfish, marlins, sailfish, they can get going pretty fast. If you watch this ribbon eel swim, you notice that it's kind of flexing. It's kind of like peristalsis, the same way a ribbon worm would swim, just bigger. And what you see is a lot of wasted energy. So there's a lot of lateral movement and not a whole lot of forward movement. And so you're not going to go very fast if you're swimming like this. So we can contrast the way an eel swims with the way a trout swims, or a sailfish or a marlin or something like that. The way you get speed is by reducing yaw, which is basically the left and the lateral movement of the head. And so if you can minimize that, you can kind of go in a straight line and reduce all the extra energy expended from going laterally. So this happens for two primary ways. The body gets a trout body versus an eel body. The trout body is going to be a lot stouter, so it's not flexible. You kind of have a stiff body and then a really flexible tail. And most of the force, the thrust, is going to come from the powerful tail. The tail is often going to be kind of upswept in a sickle shape to generate lift and power. And you're going to see this on all fast swimming animals. They're going to have a very stout body with a highly flexible tail that's probably sickle shaped, and that's what generates the force. Eels don't really have caudal fins that are large, and they just kind of have a flexible snake-like body. They're not going to be fast, powerful swimmers. This is a gigantic beast of a swordfish. Look at that guy. So the bigger they get, the faster they can swim. And also the increase in size gives you another kind of superpower, which is called regional endothermy. So endothermy is literally inside heat. And if you think about endotherms, which are colloquially called warm-blooded animals, versus ectotherms, which are cold-blooded animals, regional endothermy, fish are generally ectotherms. Their temperature is very dependent on their external environment. But large fish actually generate their own heat through the muscles of their body. So they can go so fast that they get regional warm-bloodedness that helps their oxygen diffusion rates to increase and their hearts to keep beating quickly. So all the metabolic processes are elevated in this little regional endothermy right around their core. And so swimming fast and being large has lots of benefits. Fish float. I talked about the Sarcopterygians having fat-filled swim bladders, and most teleost fish have gas-filled swim bladders, but sharks don't have swim bladders. The chondrichthyes don't. They generate lift oftentimes through their tails. So you see their tails, part of what the heterocercal tails do, and the way their pectoral fins are kind of like an airplane's wings. This generates lift. So as long as they're swimming, they're kind of keeping them, they're kind of fighting against gravity. If they were to stop swimming, they sink. But whales, not whales, this is a whale shark, which is the largest of the sharks. Sharks float by production of squalene. Squalene is a very interesting oily substance that's produced in copious amounts in the shark liver. When we dissect a shark, this will be quite obvious. The majority of a shark's internal body cavity is composed of liver because it generates so much squalene for floating. Now, if we were to peek inside a little teleost fish, so let's just peek inside, we see an air-filled bladder. And this is pretty intuitive what it's doing. You put air into it and you float. You take air out of it and you sink. But that's not it's not quite as simple as that. The air is put into the air sac by this gas gland right here, and the rete mirabile is this countercurrent exchange multiplier, which it's fairly complicated physiologically, but really all you have to know is it's pumping oxygen into the gas gland. The gas gland kind of operates as a one-way valve, and then the ovale up here can diffuse oxygen back into the bloodstream. So the gas gland controls entry, ovale controls exit, and these two things working in unison can create an incredibly high internal pressure. If you think about a steel gas cylinder filled with carbon dioxide or something, that's the amount of pressure we're talking in some of these deep sea fish inside their gas gland. So it's a pretty incredible thing that they can do. And the gas can be released through the ovale, like I mentioned, or some fish have a pneumatic duct which connects the gas gland to their esophagus. And so you're familiar with the characteristic blub, blub, blub of fish. What they're doing when they're releasing those are bubbles from the gas gland being ferried out the pneumatic duct into their mouth. So they're controlling their buoyancy when they're blub, blub, blubbing. Fish breathe dissolved oxygen in the water, and they have the most efficient system for this. Fish gills are really one of the most remarkable physiological things in nature. Their ability to extract oxygen from water is second to none. So it's still difficult to do, though. There's not a whole lot of dissolved oxygen in water, and so some larger species, like this basking shark, work breathe through something called ram ventilation, and they just swim fast enough that they force air into their open mouth across and out their gills, and this provides enough water and oxygen for them to swim. So ram ventilation is just swimming with your mouth open, getting as much water as you can. And so what happens if a basking shark stops swimming? They will drown. They won't be able to breathe. So a lot of sharks have to swim, and this is why a lot of orcas and dolphins will kill sharks by stunning them or flipping them upside down to keep them from swimming. In teleost fish that have opercula, you can see the operculum here, and when this is drawn out, it creates a negative pressure that pulls water across. And if you notice the lamellae, these are very fine little extensions, and all this is doing is increasing surface area, right? We've seen this in gills forever, from worms to insects, crustaceans, to here. If you're going to breathe, you want lots and lots of surface area. So the lamellae are responsible for that in fish. Check out the gill arch and the gill rakers. This is what is hypothesized to be the origin of those eels' second set of jaws, and you can kind of see that, right? These gill rakers offer protection from things, right? So if you're swimming in the ocean with your mouth open, you're getting all kinds of things, and you're filtering all that through your breathing apparatus. Gills have to be pretty tough. And if you notice here, the water flows counter to the flow of the blood, and this is how you get maximum oxygen absorption from the blood. Pretty amazing system. Osmoregulation is going to be especially important in freshwater fish, as you know, because freshwater fish are saltier than their environment, and so there's going to be a tendency for water to enter their body and for salt to leave their body. So they can regulate this with two different methods. They're going to counter the propensity for water to enter their body by secreting a dilute urine. So they're going to pee a lot. They're drinking a lot of water, and they have excess water, so they're going to get rid of the water however they can. But in that water, they don't want to lose their salts, and so they're going to have a tendency to lose salt to the less salty environment. And so in their kidneys, they have special salt-absorbing cells that will reuptake the sodium and all the other ions that they need, the saltiness. So they don't lose their saltiness in the fresh water. The opposite of this is in the ocean, where you often are less salty than the surrounding seawater. And so the danger there is you can actually lose water to the seawater, and you will dry out sitting in the ocean, which would be ironic, but still deadly. And so you're going to find the opposite situation in saltwater fish. Saltwater fish are going to excrete very little water, and they're going to have, rather than salt absorbing glands, they're going to have special salt secreting glands. And so if you think about this, it's not going to be too difficult. Just remember that freshwater fish are saltier than their environment, and saltwater fish are less salty than their environment. And then I think you can logically work out what the correct adaptations would be. Fish are dioecious creatures for the most part, and so males have to find females, they have to attract them, and they have to convince them to mate with them. This spectacular structure was built by, not by hand, because they don't have hands. It was actually built by mouth with a gulp of sand at a time over the course of several weeks against the flow of the ocean itself. This structure is maintained tediously by male pufferfish. And then the male pufferfish, once it's made, he'll sit and do a little dance in the middle and attract a female. Really remarkable structure. I mean, I couldn't build something like that without having to fight the ocean. But the male pufferfish will then, usually, just like with sharks, they're going to grab onto the lateral side of the female, and then fertilization is external in fish. For the most part, in sharks it was internal, but in teleost fish it's going to be external. And oftentimes there's going to be extended parental care by one or both parents. Oftentimes both parents will stay and care for the eggs. Sometimes it's just the female, sometimes it's just the male. Oh, I wanted to show you the skeleton of a pufferfish because it's just remarkable. This is what a pufferfish looks like. All the little spikes, that's what puffs out when he gets disturbed. But this little fish, I forget what the name of this fish is, but it kind of looks like a frog, so let's call this a frogfish. This frogfish, but there already was a frogfish. You can call it whatever you want, but this particular fish is a male, and he keeps the little baby eggs in his own mouth. So whenever he goes out and eats, some species don't eat at all until the eggs are hatched. Others will put the eggs down for a little while, go out and eat, and then scoop them back up. But regardless, there's often extended parental care of offspring among the fish. The last thing I want to talk about is one very strange thing that has seemingly evolved in over 7 different lineages of fish. And this is protogynous sex change. So rather than being born male or female, this is a type of environmentally determined sex. And when they're born, they're all born males. But this usually happens in social species like cleaner wrasse, like you see here, these little stripy guys here, or in clownfish or their relatives. Once they form a social group, the largest individual in that group transforms into a female. And so there'll be one female and lots of males. Now, what happens if that female dies, gets eaten, or gets old or something, or moves to a new place? Well, the second largest transforms into a female after that. Very, very strange. And this can happen the other way too. It's not always males to females. Sometimes they all start as females and then turn to males. But the sex is very fluid in a lot of these fish species. They can be born one sex and then change to another sex, and then change back to their original sex depending on the size of the fish around them, which is very strange and very unusual. Did you like the sound effects last time? I liked the sound effects last time. We'll see if we can incorporate some more of those into this here thing that we're doing. But anyway, yesterday morning, we heard the prosecution make a very compelling case that Dr. J. Cordecieras, the son of Jared Cordecieras, the rightful member of the crew, his secret was discovered by Ada Mastronomer, who was then threatening to expose his secret, and he, using his biochemical powers, manufactured an atracotoxin-like substance from local spider species, and then used that toxin to kill Dr. Mastronomer after she was injured by the tapeworm, the nematode infections, Scaris infections. So the evidence is pretty overwhelming, and most of us are leaning towards conviction one way or the other, but we let Dr. Cordecieras have his say in court, and he presented several different kinds of defense, actually. Quite elaborate. He gave it some thought, and you can tell he's quite concerned. And I mentioned yesterday that he admitted to stealing the book with the evidence of his subterfuge in it, but he still maintains his innocence about the ice cream and the navigational charts. And so far, neither the navigational charts nor any scrap of evidence about the ice cream has been uncovered as of yet. So we still can't pin that on him. And so his first attempt at defense was to say, where are the navigational charts? And just to vehemently deny that he killed Dr. Mastronomer and he stole the navigational charts and the ice cream. Also adamant about that. So he admits that he shouldn't be here. He admits that he lied about who he is, and he admits that he stole the book from Dr. Mastronomer, but he denies everything else. That's not particularly persuasive, so his second kind of defense was to kind of shift the blame, and his location of the blame he placed on Isabella, and he said, Isabella is the spider expert here. She knows and is familiar with more spiders than all the rest of us. In fact, he said he's seen her handling a venomous spider that may be the source of the atracotoxin. It's all circumstantial, no evidence of that. Isabella denied it, but he tried his best to kind of divert attention away from him to other suspects. But all the other suspects have at least one or two people vouching for them the night and time of Dr. Mastronomer's death. He was the only one without an alibi, so looking pretty weak. So next, after his first two moves failed, he pled not guilty by reason of insanity. And this got our attention because sanity is something we all hold near and dear, and on a long space voyage, we're very cautious about sanity. And what he said was that he was suffering from SADS, which is immediately made the room go quiet, because SADS is the thing that space explorers don't talk about very much. It's the demon lurking in the corners, waiting for you to stumble and fall. It's the shadow behind our backs, waiting to pounce. So we don't really talk about SADS very much. As you know, SADS stands for sudden onset acute stasis sickness, and it affects over 90% of every human who undergoes deep space hibernation. Eventually, something goes wrong during stasis. We don't know precisely if it's when they're undergoing stasis or throughout stasis or when we're waking them up, but sometime during stasis, the brain's chemistry changes, proteins start to misfold, and sometime, usually over the next 10 years, SADS starts manifesting in aberrant behavior and memory loss and paranoia, and eventually it leads to the deterioration of the brain and death. No human who has ever gotten SADS has survived longer than 9 years, so roughly 10 years is about the maximum. This is the known cost of exploring space. And for humans, this is eventually an eventual death sentence. About 90% of humans who undergo deep space hibernation will get SADS at some point, eventually within a decade. And having brought this up, he used this as a defense, saying, I'm suffering from SADS, and so my actions couldn't be, I can't be held accountable for my actions. So our expert on SADS is Vector. He is almost obsessed with SADS. He knows everything about it, all the symptoms, all the history, all the diagnoses, all the possible ways to prevent it or delay the onset. He's just obsessed with it. Well, he went through all his list of symptoms, and Dr. Cordecieras isn't suffering from SADS at all. So that defense ultimately failed, but in proffering that defense, he admitted that he killed Dr. Mastronomer and just said he can't be held accountable for it because of SADS. Bad move on his part, I suppose, because if he doesn't have SADS and now he's admitted it with all his overwhelming evidence convicting him, he doesn't really stand much of a chance. The captain is quite strict about these things. I think coming from his background, he's had a crew mutiny on him, so he has a past that he has to deal with here. But he wanted to just ship Cordecieras out into space and death sentence. We've talked him down from that to banishment, but the captain doesn't want him. He really wants it to be a death sentence. So the best compromise we could get was Snake Island. So the vote was 6-2 in favor of banishment to Snake Island. Myself and Dr. Arnold, the botanist, were the only people who voted against that ruling. I'm still not quite convinced he committed this murder. The evidence is overwhelming. I would like to find the navigational charts. That's the part of the story that doesn't quite fit. Why is he interested in the navigational charts, and why wouldn't he offer them up when at this point, right? What does he have to lose? So there are still some missing pieces, but the evidence is overwhelming for most people's. So 6-2, banished to Snake Island, where, who knows? Maybe he has the opportunity to return, in some epic, dramatic fashion, first light on the third day, as it were. Who knows? Maybe he'll be around for a sequel. It's always kind of nice to have a hermit on a mysterious island for sequels. So who knows what will become of Dr. Cordecieras in the future. Maybe he'll just die 5 seconds after stepping foot on the island. But regardless, our crew of 10 is now a crew of 8.